Dear students, this is our 20th lecture on bioorganic chemistry, and today we will start with a new chapter, that is coenzyme chemistry. And in the present lecture, we will be dealing mainly with the details of coenzyme A. Before that, we will discuss a few general concepts about coenzymes. So as I have told you in the previous lectures that many enzymes, they carry out their catalytic functions depending only on their protein structure. However, there are some classes of enzymes they which require an additional part which must be bound to the protein part, that is called as cofactors. So presence of cofactor is must for some enzymes to act under bioorganic chemistry. And these cofactors, they can either be organic compounds or inorganic metal salts like zinc 2 plus or magnesium 2 plus or manganese 2 plus ions. All these metal ions, they can also act as cofactors. And these cofactors, they are classified as coenzymes, and the prosthetic groups, depending on its nature of binding to the enzyme, that how the binding of the cofactor has taken place with the enzyme. Now if the cofactor is loosely bound to the enzyme, then these are called as coenzymes, and if the cofactor is tightly bound to the enzyme, then we call it as prosthetic groups. So in this chapter we will be dealing mainly with coenzymes where cofactors, they are loosely bound to the enzymes, and the active part of the enzyme means when an inactive enzyme is attached to a cofactor, then this is called as active enzyme and this is called as holo enzyme. So an active enzyme cofactor complex is called as holo enzyme, whereas inactive enzyme without a cofactor is called as apo enzyme. So this is how these coenzymes, they can be classified. And most commonly the coenzymes, they are vitamins, or they incorporate vitamins as part of their structure. So whole of the vitamin can be coenzyme, or part of the enzyme can be incorporated in the form of vitamin. So two examples, they are niacin and riboflavin. They are acting as coenzymes in different biochemical reactions. And vitamins, as we know, they are organic compounds which are found naturally in food, and they are essential for normal growth of the human body. And in coenzyme chemistry, the most important function of vitamins is that they act as cofactors for different types of enzymatic reactions. And most commonly occurring reactions in case of coenzyme chemistry will be oxidation reduction reactions. For example, NAD plus, which is a complex nicotine diamide, nicotinamide adenine nucleotide, which is a complex organic compound uh, derived from nicotinic acid, and this is acting as a coenzyme in different types of dehydrogenation reactions where the enzyme utilized is dehydrogenases. And these types of complex coenzymes in combination with these dehydrogenases, they help in transfer of protons between various biochemical coupled reactions. So these are some of the important substances under vitamins which are used as coenzymes. First is coenzyme A, then thiamine pyrophosphate, pyridoxal phosphate, NAD plus and NADP plus, as I have told you, FMN and FAD, lipoic acid, and cyanocobalamin. So all these are the important substances which act as coenzymes in various types of biosynthetic reactions. So we will discuss these one by one. In the present lecture, we will be discussing mainly the chemistry of coenzyme A. And these are some of the important coenzymes, their function, and the vitamin from which they are derived. For example, if coenzyme is biotin, then the function, the vitamin is vitamin H, and its function is carboxylation. So this coenzyme in combination with the enzyme will be used for carboxylation reactions. Similarly, if coenzyme is tetrahydrofolate, then vitamin is folate, and the reaction which will take place is one carbon transfer. So they will catalyze as a coenzyme one carbon transfer reactions. Then if vitamin is lipoate, then coenzyme can be lipoate and dihydrolipate. And if coenzyme is lipoate, then this will catalyze the oxidation reactions. If it is dihydrolipate, then it will catalyze the reduction reactions. Similarly, nicotine adenine dinucleotide NAD plus or NADP plus, that is phosphate of NAD, or NADH or NADPH. If these are the coenzymes, then vitamin from which they are derived is niacin, and they also undergo oxidation reduction reactions. Then if coenzyme is coenzyme A, then the vitamin is pantothenic acid, and this is used for transfer of acyl group in various biochemical reactions. Then pyridoxal phosphate, vitamin is vitamin B6, and this is used for decarboxylation reaction, transamination reaction, and racemization reaction. So all these three types of reactions, they are catalyzed by pyridoxal phosphate as coenzyme in combination with some enzyme. And vitamin part is vitamin B6 in this case. Similarly, FAD and FMN, this will catalyze oxidation reactions. FADH and FMNH2, they will catalyze reduction reactions. And these coenzymes, they are part of vitamin B2, that is riboflavin. Then next is vitamin B1. The coenzyme is thiamine pyrophosphate, and this catalyzes two carbon transfer reactions in case of biosynthetic reactions. And last one is coenzyme B12. This is derived from vitamin B12, and this is used for isomerization reactions in biosynthetic chemistry. Now, coenzyme A is derived from pantothenic acid, which is also known as vitamin B5. So, this is the structure for vitamin B5, pantothenic acid, and this pantothenic acid is part of coenzyme A. And In addition to pantothenic acid, we have two additional components present in the coenzyme A. One is a phosphate derivative of ADP, adenosine diphosphate. So this is the structure of phosphate derivative of ADP. This is bound to the CH2COH part of the pantothenic acid. So OH from here and H from the phosphate group, water is removed, and we will get a carbon oxygen bond like this between adenosine diphosphate and pantothenic acid. And similarly, other additional component is beta mercaptoethylamine. So this beta alanine is joined by beta mercaptoethylamine. So amide linkage, this carboxylic acid part will form an amide linkage with the beta alanine, and then carboxylic acid of the beta alanine will form an amide linkage with the beta mercaptoethylamine. So this whole part of the molecule is called as coenzyme A, where we can say that we have these four parts: beta mercaptoethylamine, beta alanine part, pantothenic acid, and adenosine diphosphate. And this pantothenic acid is the vitamin B5 part of the coenzyme. And coenzyme A is involved in transfer of acyl groups in various types of biosynthetic reactions, and these reactions they are important for release of energy from carbohydrates. Then they are important in the synthesis of glucose, then synthesis and degradation of fatty acids, they are also important. Then synthesis of steroids and steroidal hormones, these reactions are important. Then synthesis of adrenocortical hormones and cholesterol is also governed by the reactions involving coenzyme A. And a number of proteins have also been modified by attachment of long chain fatty acids, which are transferred from coenzyme A, and these modifications are known as protein acylations. And these protein acylation reactions, they play a central role in cell signaling, for which we will see some examples in the subsequent slides. Now, biochemical functions of coenzyme A, as you know, this is the basic structure of coenzyme A, and this thiol group or free sulfahydryl group at the end is used up for various types of biochemical reaction. So this will react with the organic acids such as acetate to form a thioester bond, and this thioester, they will be used up in transfer of the acyl group to other components. So that is what I have said, that coenzyme A is a carrier of acetate and other acyl groups, and acetyl group transferred to coenzyme A by enzymatic reactions forming acetyl coenzyme A. Acetyl coenzyme A is a high energy and activated form of coenzyme A, and this acetyl coenzyme A can be generated by oxidative decarboxylation of pyruvate or stepwise breakdown of different types of fatty acids. The role of coenzyme A in metabolic processes can be in TCA, that is tricarboxylic acid cycle, and beta oxidation pathway for activation of fatty acids. Now, for example, in TCA cycle, we know that glycolytic pathway ends in the formation of pyruvate. And then this pyruvate is decarboxylated uh, to form acetyl coenzyme A, and then initiation of the TCA cycle takes place. So you can see over here that coenzyme A reacts with pyruvate in the presence of NAD plus as uh, the another reactant which is converted into NADH with the transfer of hydrogen. So basically this will undergo decarboxylation and this acyl group will be transferred to coenzyme A and we will get acetyl coenzyme A and carbon dioxide will be formed. And this reaction is catalyzed by pyruvate dehydrogenase enzyme which is a complex enzyme which consists of three enzymes, five coenzymes and two regulatory proteins. So this complex enzyme pyruvate dehydrogenase catalyzes the decarboxylation of pyruvate to for the synthesis of acetyl coenzyme A in the TCA cycle. Then similarly, activation of different types of fatty acid can take place, and we will get fatty acyl coenzyme A, which is a high energy compound from coenzyme A, and this reaction is catalyzed by acyl coenzyme A synthetase. So basically what happens is that coenzyme A in presence of this enzyme acyl coenzyme A synthetase will react with the fatty acid in presence of ATP, and ATP will be converted into AMP and diphosphate, and energy will be released. That energy will be used up in formation of a thioester bond between SH and this OH. So OH from here, H from here is eliminated and we will get this acylated fatty acyl coenzyme A as the product. Similarly, acetate can be transferred to coenzyme A by using this acetyl coenzyme A synthetase as the enzyme in presence of ATP. The same way, OH from here, H from here, water will be eliminated and acyl group will be transferred to this coenzyme A and we will get acetyl coenzyme A as the product, which is a high energy compound and which can be used for transfer of acetate in biological systems later. Similarly, coenzyme A is also involved in biosynthesis of phospholipids. So what happens is here substrate is dihydroxyacetone phosphate. This hydroxyl group of dihydroxyacetone phosphate will act as a nucleophile in presence of the enzyme dihydroxyacetone phosphate acyl transferase. So active site present on this enzyme as a base will abstract this proton on the hydroxyl group, and this oxygen will then act as a nucleophile and will attack the acetyl or acyl coenzyme A carbon, and we will get this type of tetrahedral intermediate will be formed between coenzyme A and the substrate. So substrate coenzyme complex will be formed, which is a tetrahedral intermediate, and then now this active site has become acidic in nature. This proton will stabilize, this will act as positively charged body, and this will neutralize this negative charge on the tetrahedral intermediate, and then this will form a carbon oxygen double bond, and coenzyme A will be eliminated, and formation of a phospholipid will take place, and ester leakage will be formed like this. So this is how the dihydroxyacetone phosphate will be converted into a phospholipid. So basically reaction is this OH part, and this acyl part will form an ester linkage. Similarly, biosynthesis of acetylcholine, this is also catalyzed by acetyl coenzyme A. Acetylcholine is a neurotransmitter which transmits nerve impulses across the nerve cells, and this acetylcholine is synthesized from the reaction of acetyl coenzyme A and choline, and this acetylcholine needs to be hydrolyzed very quickly after the transmission of the nerve impulse between the cells. So this hydrolysis takes place in the presence of enzyme acetylcholine esterase, which will hydrolyze this bond, and we will get choline, and acetate will be formed as another product of hydrolysis. So here for the formation of acetylcholine, acetyl coenzyme A will react with choline, and H from here, and S negative coenzyme A from here, they will form coenzyme A, and acetyl will be transferred from acetyl coenzyme A to choline, and it will become acetylcholine, and which will be used for transmission of the nerve impulses. Then the activity of this enzyme acetylcholine esterase is due to this group. This is proved from the fact that when this enzyme is reacted with diisopropyl fluorophosphate, that is DIFP, military nerve gas, which is known as. So what will happen? H from here and F from here. HF will be eliminated, and this enzyme will form a product. Enzyme substrate complex will be formed, covalent complex will be formed, which will further inhibit the activity of acetylcholine esterase. So when such a permanent inhibition of the enzyme activity can take place, that can only take place if this is the active site of the enzyme which is involved. So this reaction proves that in case of acetylcholine esterase, the active group is this one, CH2OH group, and this undergoes inhibition when it reacts with DIFP. Similarly, one important reaction is conversion of acetyl coenzyme A into malonyl coenzyme A. So this can be achieved from the reaction via carboxylation reaction, and here the cofactor used is biotin, and the enzyme used is acetyl coenzyme A carboxylase. So what happens is acetyl coenzyme A reacts with bicarbonate and ATP in the presence of biotin as cofactor, and magnesium 2 plus metal ions, they are also present as cofactors, and acetyl coenzyme A carboxylase is acting as the enzyme, and this malonyl part will be added to the acetyl coenzyme A, and we will get malonyl coenzyme A as the product. And the coenzyme biotin is linked to the active site of the enzyme which have been used, acetyl coenzyme A carboxylase, like this. So this is biotin part, and this is lysine, which is an active site present on the enzyme acetyl coenzyme A carboxylase. So this will bound to the enzyme to give us enzyme coenzyme complex, and this enzyme coenzyme complex is used up for conversion of acetyl coenzyme A into malonyl coenzyme A. Then this acetyl coenzyme A can be used for conversion into different types of terpenes or terpenoids. This 2-carbon acetyl group of acetyl coenzyme A is the building block for all types of steroids and terpenoids. And these two main reactions they take place during the formation of terpenoids from acetyl coenzyme A. One is aldol condensation and other is collision condensation. And uh, the first step in these types of reaction is formation of an isoprene unit, which is a 5-carbon unit, which act as we can say monomer for the synthesis of different types of terpenoids. And this 5-membered carbon unit, isoprene unit, is used for synthesis of this mevalonic acid, which is a 6-carbon compound. And this mevalonic acid 3R form act as precursor for different types of isoprenoids or terpenes as the compounds. And these terpenes, they are generated by conversion of acetate into branched chain intermediate, that is isopentyl pyrophosphate, which is the biological isoprene unit from which all types of terpenoids they will be synthesized. For example, in the first step, two molecules of acetyl coenzyme A, they will undergo collision condensation in the presence of this enzyme acetoacetyl coenzyme A thiolase, and we will get this acetoacetyl coenzyme A, and one molecule of coenzyme A will be eliminated from here. Then this acetoacetyl coenzyme A will react in the presence of enzyme 3-hydroxy 3-methyl glutaryl coenzyme A synthetase, where another acetyl group will be transferred from acetyl coenzyme A, which will become coenzyme A, and acetyl part from here will be transferred to acetoacetyl coenzyme A, and we will get HMG coenzyme A like this. This is S conformation of beta hydroxy beta methyl glutaryl coenzyme A. Then this in presence of HMG coenzyme A reductase, reduction of this will take place, and a molecule of coenzyme A will be eliminated, and this will be reduced to CH2OH, and we will get mevalonic acid as the product of this reduction in presence of NADPH. And then this R mevalonic acid will act as a precursor for the synthesis of IPP, that is isopentyl pyrophosphate, which from which all these terpenes they will be synthesized. Uh, in the first case, R mevalonic acid will react with mevalonate kinase enzyme, and phosphorylation of this OH group will take place, and we will get a phosphate of this alcoholic functional group. Then in the presence of this enzyme phosphomevalonate kinase, another phosphate group will get attached to this, and we will get diphosphate of this mevalonic acid. Then mevalonate 5-diphosphate decarboxylase will convert this into IPP by decarboxylation followed by dehydration, and this in the presence of this enzyme this will be converted into IPP, decarboxylation of this and dehydration at this place, H from here, OH from here, we will get this IPP as the product, which will act as a precursor for the synthesis of different types of terpenes. And this IPP can undergo isomerization in the presence of this IPP isomerase enzyme to this more stable form, dimethyllyl pyrophosphate DMAPP, and this whole process is called as mevalonic acid pathway for the synthesis of terpenes. Then like this, different types of hemiterpenes, monoterpenes, sesquiterpenes, diterpenes, triterpenes, and tetraterpenes, they can be formed from the reaction of IPP with DMAPP. So when one molecule of IPP and one molecule of DMAPP, they combine with each other, we will get geranyl diphosphate as the product with the elimination of one diphosphate from these two. And this geranyl phosphate is an example of monoterpenes. Then this geranyl diphosphate on reaction with one more molecule of IPP with the elimination of a diphosphate is converted into farnesyl diphosphate FPP, which is your sesquiditerpenes. Then this FPP will further react with one more molecule of IPP, and this will be converted into GGPP, geranyl diphosphate, with the elimination of one diphosphate molecule. And this is our example of diterpenes. Similarly, one molecule of FPP when combined with another molecule of FPP, this can be converted into sequeline, which are example of triterpenes. If two molecules of GGP they condense with each other with the elimination of diphosphate, then we will get phytone as the product, which are example of tetraterpenes. So this is how these precursor IPP or DMAPP they condense with each other in different manners to give us different types of terpenes: hemiterpenes, monoterpenes, sesquiterpenes, diterpenes, triterpenes, tetraterpenes. Then the mechanism for the biosynthesis of monoterpenes. The monoterpenes basically they require a nucleophilic attack of the carbon-carbon double bond at this place with the elimination of this H positive from here, and this will IPP will act as a nucleophile to this DMAPP, and we will get this type of uh, product with the elimination of this diphosphate. So this will act as a leaving group. This will act as a nucleophile, and basically we can say this is a nucleophilic substitution reaction. And either this can take place by ion pair method. So here this will act as a nucleophile, and oxygen is highly negative, delta negative charge, so carbon will be having delta positive charge. So this is ion pair method. Or bimolecular pathways, that simply this will act as a nucleophile and this will be substituted. So nucleophilic substitution reaction. So this is the mechanism that how a molecule of IPP and DMAPP, they will give us monoterpenes as the product. So these are the different types of terpenes which can be formed in different ways from this condensation of IPP and DMAPP. We can get nearyl pyrophosphate, then with the movement of this double bond elimination of this phosphate, we will get this type of carbonium carbocation. Then this carbocation, this double bond will form a carbon-carbon bond and positive charge will come to this place. And from this type of carbocation, either by reaction with water, we can get alpha terpenols, or this can further undergo rearrangement reactions to give us alpha pinene, beta pinene, phenchone, isobornylene, camphor. So all these reactions they take place by Wagner-Merwin rearrangement. And Wagner-Merwin rearrangement we have already done, that this is a carbocation type of rearrangement which takes place in case of natural products. So through Wagner-Merwin rearrangement, these carbocations they can be converted into different types of natural products, like we have shown over here: thuzane, terpenol, phorol, camphene, camphor, pinene, terpenol, beta pinene, phenchone. So all these types of natural products they can be formed through Wagner-Merwin rearrangement. Then role of coenzyme A in metabolism. Uh, we know that metabolism can be divided into two types: catabolism, where complex molecules they are converted into simple molecules and energy is released, or anabolism, where simple molecules they take up that energy and they are converted into complex molecules. So the role of acetyl coenzyme is central in catabolism as well as in anabolism. So catabolism, you can say that the proteins they are converted into amino acids. So proteins they are complex molecules and amino acids they are simple molecules. So energy will be released. That energy will be used up in reaction of the pyruvate to convert this into acetyl coenzyme A through decarboxylation reaction. Then carbohydrates they can be converted into monosaccharides. Monosaccharides they are simple molecules, carbohydrates they are complex molecules. Fats they are complex molecules, fatty acids they are simple molecules. And energy released during this catabolism reactions they will be used up for conversion of pyruvate into acetyl coenzyme. Then this coenzyme A can be used for detoxification. Then ketones they are converted into energy. Fatty acids they are converted into triglycerides, because fatty acids they will react with acetyl coenzyme A and acetyl part will be transferred to the fatty acids. Similarly, cholesterol can be converted into vitamin D, steroids, and hormones through these cannibalism reactions using acetyl coenzyme A. So, this is all about coenzyme A. Thank you very much.